And our next presentation, we will hear from John Mojica and Robert Bremer, multi-year assessment of land-based sources of pollution and water quality in Biscayne Bay, Florida, for our next 15-minute presentation.
Hello, everyone. Thanks for joining us and be here. Again, first of all, let me introduce myself. I'm John Mojica. I'm here with Robert Bremer. We are researchers at the University of Miami, at the CIMAS. Also, we are NOAA affiliated. And here we are going to talk about this project in Biscayne Bay. We call it multi-year assessment of land-based source of pollution and water quality in Biscayne Bay. We basically create a multivariable analysis of the stations that we have been collected, in general 20 stations. I'm going to start with our takeaways that I guess that is quite important. And first of all, that we identify the primary source of pollution or pollutants in the area that are Miami River, Little River, and the Miami Central outfall, and all talking about the North Biscayne Bay. As you can see this photo, I don't know if anybody of you have been in the area. This is the outfall that is around 4 miles away from the coastline, and it's quite flat at some point, bubbly and smelly, but well, we can identify that something is happening here, and also that's why we identify that there is a bacterial display, particularly a hotspot near this area and also the Miami River. We identify that also there are some kind of concerns about the nutrients and how the trends of those ones are increasing in these areas. And of course, we encourage all of you to keep with the monitoring. And here in general, these are the stations over the North Biscayne Bay. We used to talk about inshore and offshore, but for you, the inshore, the red dots, are located over the near the urbanized area in Miami, North Miami. And the yellow ones is that we used to call offshore, are closely located over the government cut. And all those in total, we have 20 stations, and we used to collect the samples of the inshore stations after the high peak of tides, to record what is happening with the land-based pollutants, and the offshore ones after the low tide, just to identify the oceanographic or get the signature of the oceanographic input. Our methodology over all those 20 stations, we used to take measures at the surface and at the bottom using a multiparametric sensor, taking bottles, all the samples, and recording water properties and water quality parameters in the area. We proceed with our general QC. Just to keep a good quality of all the variables. Here we develop a principal component analysis where we identified two main components. The first one is about the nitrogen and the silicates, that those variables are quite related with canal discharge and storm waters, so we can identify that those variables are related with terrestrial runoff. And for the phosphates and the ammonium, we identified that is also another principal component, and these ones are more related with septic tanks and everything related with wastewater. In general, variables such as temperature, salinity doesn't have a relevant weight here, so far just those two principal components. Here, over these three main areas, we create a hierarchical clustering that helps us to identify or classify the level of how homogeneously will going to be the bay, and in this case it's not homogeneously at all. And we can identify that in general all the stations, most of the stations that are located near the coast are quite related over his variables, and the ones of course over the near the government code, all this area are more related between each other, all the different variables. Just I want to mention how this Miami River, Little River, and the government code, all these three dots are at some point quite parenty all the other stations or the composition in the area. About the temporal, the trends that we can identify. One of the relevant one, as mentioned by Edward and this morning by Alexander, was about the phosphates that so far data from September 2021 up to December 2025, we have this high trend of accumulation of phosphates in the area. This is just the plot for the Miami River over this area, but in general we can identify that trending that spread all over the Biscayne Bay in these areas. Here also another of the variables that we identify is the silicates, where we identify that are decreasing at some trend, but still is higher, and those ones are located over here. About the distribution of these plumes, we can identify about the silicates and the nitrates, and that there is a general gradient that coming from the land to the ocean park as something that we identify and are quite related with the salinity distribution. Also something about the second principal component, about the phosphates over the wet season, here phosphate dry, ammonium wet and dry season, that there is a clear hotspot that we can identify, and this one coming literally from the outfall. About chlorophyll over the wet and dry season, also there is some gradient that coming from the west to the east, and if we relate with all the other variables are quite related, that can we start a loop of eutrophication, something that we have to take care.
Thank you, John. Again, my name is Rob, and I'm going to hop in with the bacterial component of this analysis. So we use a methodology called microbial source tracking to amplify specific gene segments that can tell us what species of bacteria are in the water during that sample. And based on what species amplify, we can tell a lot about the type of pollution that's occurring. So in this project, we use 3 different markers. First off, we have human-associated bacteroides. This suggests that the pollution is anthropogenic in nature. It's really critical for identifying point source pollution, like failing septic systems, leaky sewers, or wastewater treatment plant issues. Second, we have a DG3 marker for canine-associated bacteroides. This is typically used for non-point source pollution, and it can inform more general social issues or infrastructure problems like overwhelmed stormwater systems. And thirdly, the marker NRO1A tells us about the level of enterococci in the water. This is a good all-around indicator of the biological health of the ocean, and is often indicative of other disease-causing agents in the water. So, moving on, human-associated bacteroides. We found that the levels are pretty stable throughout the 4-year monitoring period that we're presenting today, and it's actually decreasing a little bit over time, which is great news. This may be due to the fact that we've had like a pretty dry last 2 years, or it may be due to the city making improvements. We don't really know. Hopefully that'll come out further in analysis. But we did find that there were a couple problem sites, notably the outfall sites, and all the outfall sites were really high, these pink, purple, and navy, and Miami River in black was also very high, which are sort of understandable. This is an anthropogenic marker. These are the anthropogenic sites. Miami River is in the center of Miami, and the outfall basically is accumulating everything that we take as a city and dishing it out to the ocean. Our dog-associated bacteroides was pretty stochastic in nature, which is, I guess, a good thing. Not a lot of our samples did amplify or didn't amplify in detectable quantities, but most of our spikes or amplifications were in basically areas with recreational activity in dogs, like you might imagine, and you guessed it, the outfall also had pretty high levels of this bacteria. And thirdly, the enterococci values were had a lot more fluctuations seasonally, and this red dotted line is a recommended beach action value of 158. A lot of our samples were over that value. This may have something to do with the residence time of enterococci, but it is a little concerning. more to these fluctuations, they appear to be semi-seasonal. We know that rainfall contributes a lot to these spikes, but it doesn't quite match up with the wet seasons in Miami here in blue. They don't quite account for the fact that some of these spikes are in the dry season, some are in the wet season. But if we overlay rainfall, we can actually learn a lot about, why these are spiking. If you see some of these periods where it's been dry for a little bit and then rains again, that's when we get a huge spike of enterococci here in like 2023 and 2024. Right. So just some key takeaways from microbials. There's a lot of concentration of human-associated bacteroides in Miami River and the outfall sites. For DG3, it isn't a pervasive issue. Detection is more localized and stochastic and occurs at specific sites. And for enterococci, these are frequently high in most of our samples and strongly influenced by seasonality and rainfall patterns. So as we move forward as a city with rising sea levels and aging infrastructure, we should keep all of this in mind. And then just to reiterate some of John's points, there were two independent drivers. We kind of have the runoff accounting for nitrogen and silicate, and more point source issues for phosphorus and ammonia. There's also different zones, so the more anthropogenic zones, the outfall, Miami River, Little River, should be treated a little bit differently than our inshore and offshore sites that aren't those sort of problem sites. Also, we should look out for eutrophication with rising phosphorus, because that can lead to the seagrass that we all love and care about. And there's peak stress in the wet season with a lot of that thermal load, low dissolved oxygen, and a lot of runoff. Thank you, everyone.